What are the basic differences then between PGD and PGS? Because patients use these terms interchangeably and they're really not the same thing. With preimplantation genetic diagnosis, PGD, we have typically a couple, both members of the couple are carriers of a specific genetic disorder. So classic example, African American couple, they both have sickle cell trait, which means that of the two genes that code for hemoglobin, one of those two genes is abnormal, and they both have that same abnormality. Therefore, in the woman, half of the eggs that she makes will have the normal hemoglobin gene, and half of the eggs will have the abnormal gene. Same thing in men with sperm. Half of the sperm they make will have the normal hemoglobin gene, the other half will have the abnormal gene. So here's the problem. When the sperm and the egg combine, if the normal egg is fertilized by the normal sperm, now you have a baby that does not have sickle cell disease, nor is it a carrier of the gene. So nor does it have sickle cell trait. But on the other hand, if that baby inherits the abnormal copy from the egg or the abnormal copy from the sperm and a normal copy from the other one, that baby then has sickle cell trait, which can be still a very problematic disorder. Now, if that baby, or if that embryo, inherits the abnormal gene from the egg and the abnormal gene from the sperm, now that baby has sickle cell disease, which is a life-threatening abnormality lifelong. So with preimplantation genetic diagnosis, we're not looking at the individual chromosomes, we're looking at the genes within those individual chromosomes. But the rub is that we have to know exactly what we're looking for. Because there are between 5 and 25,000 genes per chromosome, it's not technically possible today to screen for everyone. Preimplantation genetic screening, on the other hand, is very different. We still have to go through in vitro fertilization, do the biopsy, freeze the blastocysts, send those cells off for biopsy. But now what we're looking at is we're looking at the 22 pairs of chromosomes and the 2 sex chromosomes. 2 X's if it's a female, an X and a Y if it's a male. And we are counting those chromosomes to make sure that we have the correct numbers of copies in that embryo. So for example, classic example, Down syndrome, an extra chromosome number 21. So instead of having 46 chromosomes, that embryo has 47 chromosomes, the extra one being a number 21. So we can screen embryos for all kinds of abnormalities in the number of chromosomes, that's called aneuploidy, and then we can determine which embryos are normal, which ones are abnormal, and we can transfer only the normal embryos. This results in higher pregnancy rates, higher delivery rates, lower miscarriage rates. And in fact, research in our program has shown that this is cost effective for all couples going through in vitro fertilization.